Hello and welcome back to Excelsior Academy. So today we'll be doing RNA splicing. So far we have completed with the transcription. So next thing, like once your RNA is ready, okay. So to go on to the next step, that is a protein synthesis. Before that, we need to make sure that your RNA is ready and it is free of interons. Okay, so that is what today we are going to discuss, like what are the different ways by which the RNA is getting spliced so that we get different combinations of RNA product or the transcript which will be further translated to protein. Okay, so this is the basic mechanism by which the RNA is getting spliced. So here you can see that you have got the DNA, it gets transcribed, you have got the 5' leader and the non-coding region, right? Remember, we have talked about 5' UTR region and the 3' UTR region, that is untranslated region, correct? Now, after the splicing, what happens is that all your interons are removed and only the exons are taken. Okay, now this is your transcript ready. It's your ready transcript, okay, or the spliced mRNA we say, but it is retaining the 5' and the 3' UTR. Now this is important because it carries the sequences which is required by the ribosome so that they can interact. ठीक है, इसलिए ये 5' and 3' UTR regions are required. Otherwise, see here itself we know that the first codon what you are going to have is AUG. So इससे पहले it needs to recognize the ribosomes properly. It has to align, and that's why these regions are important. Okay. Now, this is the graph which shows like this is representing the average number of introns per gene. Okay, so here you can see that as the diversity increases, okay, or the complexity increases of the organism, the number of interons per gene we are talking about. Okay, so you can see that in the case of humans, okay, this is last one jo hai Homo sapiens, this is for your human scenario. You can see that it has got the maximum. So maximum number of introns it has per gene. So if I say clearly, agar hum ise thoda sa tally kare, so we can see that around 7 to 8 introns are present in one gene. Okay, in one gene in the case of the humans. Now, if you see, now let us see all the red cases where you can see all those red highlighted ones are there. Okay, now looking at these red names, what can you tell about it? क्यों ऐसा लगता है कि उसको उन्होंने red में कर रखा है? Is something wrong with that? Can you just look over the names and guess क्यों it is highlighted separately? So first one is Saccharomyces cerevisiae. Okay, then next is, this is one. The next is Drosophila melanogaster. Next, उसके बाद क्या है? C. elegans, that is the Xenozaptus elegans. Next, Arabidopsis thaliana. तो ये नाम सब सुन के क्या लगता है? And then of course M. musculus. This is your mouse model. Okay, so ये जो highlighted वाले हैं, it is all representing you that they are your experimental model. Okay, they are experimental model. So you know that अगर हम मुझे yeast में काम करना है, so we have got so many varieties of so many species of yeast, right? But we always go with the Saccharomyces cerevisiae. Fruit fly में सिर्फ Drosophila melanogaster में ही जाते हैं. Nematode में C. elegans जाते हैं. Plants में Arabidopsis thaliana में जाते हैं. And if suppose I want a mammalian model, then I go for the musculus. Okay, mouse model I go. Okay, clear? Now look at their gene diversity. So here, तो यहाँ पे देखो, it's hardly any introns are present per gene. Okay, now once I go to the Drosophila melanogaster, at least 3 are there per gene. Now C. elegans, around 4 to 5 it is there. Then Arabidopsis thaliana, around 6 to 7 is there. And now if I go to mouse, I have got around 7, and humans में और ज़्यादा हो जाता है. ठीक है? So as the diversity of the complexity in the organism increases, the number of introns per gene is also increasing. Clear? Is this figure is clear?
Yes.
Very simple. Yeah.
Yes, ma'am.
Okay. So there are some pre-mRNAs which can be spliced in more than one way, and it is also known as alternate splicing. Okay, this is a very beautiful concept, and you need to remember this concept because this is going to come when we are going to do the immunology part also. It is very important that time. Okay, so this thing, it should be very clear to you, like alternate splicing kya cheez hai. So alternate splicing is like, if suppose you have got one, let us say that, okay, let us say that these are your exons, okay, and these are your introns. Now, alternate splicing says that you can have the product formation, that is your transcribed or the spliced RNA can be formed in different ways. So I can have combination of 1, 2, 3 exons. I can have 1, 3, 2. I can have, let's say, 2, 3, 1. different combinations I can have. समझ रहे हैं? So it means what? 1, 2, 3 ऐसे together they will come. And sometimes 1 will come here, 3 will come first, and then 2 will come. So depending on the requirement, the same gene, okay, same gene can give you different or the alternate mRNAs, which will be coded to give you different proteins. And obviously these alternative products will be known as isoforms. समझ रहे हैं? Okay. Have you heard about the VDJ recombination with respect to immunology? कभी सुना है क्या VDJ recombination? Ever heard about this?
No.
नहीं? No one? Okay, so it's okay. I mean, it's not that at this point it is not that crucial. The thing is that this VDJ recombination, okay, this VDJ recombination is important as part of the immunology. Okay, so this is actually a mechanism of somatic recombination. Now this helps in the formation of your antibodies. So you know that we have got different antibodies, right? So it is going to give us different combinations of the antibodies. And for example, the immunoglobulins, the antibodies are nothing but your Ig only, right? Immunoglobulins. So they will have different representations of these globin molecules. So that all is done by this VDJ. Now VDJ kya hai? VDJ is nothing but they are the genes. Okay. So VDJ, where V stands for the variable, D stands for the diversity, and J stands for the joining. So if you know in your antibodies, we have something like this, okay? Oh, sorry, 1 second. So V, D, and J. So antibodies are like this, okay? Heavy chain and light chain you have got, right? Now these heavy chains, light chains, they all are like made up of these. So you have got the variable region, which is deciding what kind of antigen it should bind, then diversity it is there, and the joining genes are present. Okay, so this different recombination or different combination of these genes, okay, the arrangement of these genes will help in formation of different antibodies. So अगर आप मैं ये बोलती हूँ that I have got IgA antibody, so IgA में you will have different diversity because this IgA will be having differences in its variable group so that it can bind to several thousands of antigens and help me in the protection. थोड़ा सा समझ में आया? Of course, we will be doing in detail once we start the immunology. But is this much concept is clear? Like alternate splicing is responsible for your VDJ recombination, which is nothing but resulting in different expression of your antibodies. ठीक है? So where variable, diversity, and joining. So these are the genes. Itna clear hai? So that's why if I have got IgA of different diversity, those will be known as the isoforms. Okay. Is the concept of alternate splicing is clear?
Yes, ma'am.
Okay, now let's go to the chemistry of RNA splicing. So this topic of RNA splicing, it will take 2 classes. So by maybe in the next class, we'll complete this. Okay. Okay, now coming to the chemistry behind. So as you can see over here, this is one example. So in this figure, if you look carefully, it is based on the sequences which are present at the intron exon boundaries. Okay, and it is like universal. So the most highly conserved, okay, first of all, it happens only in eukaryotes. the RNA splicing. Okay, why? Because in the prokaryotes you don't have introns, so there is no need of having RNA splicing. Correct. In the case of eukaryotes, only introns are present, and that's why you need the RNA splicing before it goes for the translation. Now the information is present in the intron exon boundaries, and that information is nothing but the sequence. Okay. Now look at the sequence. So at the end of your 5' exon, जो आपका इस side present है, whichever the exons are present over here, at that you have got the sequence known as 5' splice site. Okay. Then in the intron you are going to have A. Okay. Then again in the 3' splice side you are going to have this sequence. Okay, so it is the most highly conserved sequences, which is the GU in the 5' splice site and AG in the 3' splice site, A in the branch side. Can you just remember? Simple, right? So 5' splice site में GU, 3' splice site में आपका AG and branch side में A. इतना clear है? So this is what you need to remember. I'll just repeat once again. So here in the chemistry, like how does RNA splicing happens? The information is present in the sequences present at the intron exon boundaries. Now this is the boundary. So this is your exon ka boundary. Exon. This is exon. This is starting of intron. Correct. Now at this boundary, it is known as 5' splice site where you have got the GU. Then in the sequence in the middle in the intron, you have got A. Now it is called branch site because this particular sequence will get detached from here, okay, and will bind over here. Then at the 3' splice side, you have got AG. Clear? Itna clear hai? Now, when I say that it gets detached, what does it mean? So in the RNA, there is a phosphodiester bond, correct? It's a phosphodiester bond. So in the process of RNA splicing, there will be breakage of 2 phosphodiester bond and formation of 2 phosphodiester bond. So the net result is 0. अब कैसे है मैं बताती हूँ. So your first bond is broken over here. That is G के बीच में. Okay. So what will happen? This G, this free G will go and form a bond with A. Okay. This is your first bond. Up second is this bond will be broken over here. Okay, this is your second phosphodiester bond is broken, and here the G will go and bind with this G. So यहाँ पे break हुआ, यहाँ पे बना, यहाँ पे break हुआ, and यहाँ पे इन दोनों का बना. So net result 0. So 2 phosphodiester bond break हुए हैं और 2 phosphodiester bond बने हैं. Guys, clear? I mean, it will be clear in the upcoming slides. Don't worry. Okay. So the intron which is removed, it is removed in the form of lariat as the flanking exons are joined. So look at over here. Now look at this figure. Okay. So you can see it over here. So first one, so A ka O2-OH is present. Okay. So intron is removed through 2 successive trans esterification reactions in which the phosphodiester linkages within the mRNA are broken and new ones are formed. Okay. And the process is known as trans esterification. So the first reaction is triggered by 2-OH of the A. ये आपका क्या है? Branch site है. ठीक है? So this is going to attack over here. Now, please remember, whenever we previously also when we were talking about DNA polymerase during the replication, RNA polymerase during transcription, it was all your SN2 mechanism. Okay? So here also it is your SN2 mechanism. Now, so this group will act as a nucleophile and it will attack the conserved G in the 5' splice site. So now what is happening? You can see this 2-OH has attacked over here and another phosphodiester bond was formed. So you can see this group is formed. Okay, ये GP ये आपका guanine है, उसका phosphate इसके साथ bond बनाया OH के साथ. Usually it is always the 3-OH and 5' phosphate, right? But in the intron it is 2-OH which is which is attacking over here and forming a phosphodiester bond. Correct? Now this OH 3-OH is free. This will go and attack at this phosphate. So there is a breaking of the phosphodiester bond and formation of another phosphodiester bond. Okay. So as a result, okay, after this two trans-esterification reaction, what I'm going to get is this gets free from here. So this is your intron lariat, and this is your splice exons, which is ready. Clear? So you can see that two phosphodiester bond was formed and two were broken. Clear, everyone? Shruti, you have just joined now, but I hope this slide is clear to you.
Yes, ma'am. Yeah.
Okay. What about others? Tanvi and Akshaya, are you able to follow? See, once you understand this, then only जो आगे हम पढ़ने वाले हैं वो clear होगा. Otherwise, it will become difficult then. अगर कोई doubt है, we can solve it now itself. Yes. Anything you want to ask, Tanvi and Akshaya?
No, ma'am. It's clear.
Okay. Okay. Chalo. Okay, so this is just the structure, that three-way junction formed during the splicing reaction. So if you see carefully, okay, so this is your 5' end of the interon, जो हम अभी बात कर रहे थे, उसका phosphate and उसका G, correct? If you can understand, and वहां से आता है आपका दूसरा 3' end of intron. So this is how the structure will look like when it is attacking, okay, during the splicing reaction. But that's okay. I mean, you don't have to study the structure, just understand how it is happening. Okay. Now, that was the basic mechanism behind the RNA splicing. Now, how this splicing is done? किसी को तो help करनी पड़ेगी ना, lariat बनाने में, वहां से 5' and 3' तक लाने के लिए. So this is all done by the spliceosome proteins. Okay. So this is also known as spliceosome machinery. This is the most important ones. And it consists of having around 5 RNAs which are present. Okay, this together they are known as small nuclear RNAs. So they are nothing but like they are the ribonucleoproteins. Okay? Why? Because they have got the RNA as well as the proteins which are associated with them. Now, if you think why this spliceosome is having both RNA and proteins, so the proteins part is actually playing the role of the enzymes forming the active site, and the RNA part is required so that it can form a complementary base pair with the mRNA. Okay, and to bring the catalytic site, like for example, एक के साथ जब वो base pairing करता है, it becomes easy for the enzyme to hold and break. Okay, so there are figures, from that it will be clear to you. So each of these RNAs which is present in these spliceosomes are around 100 to 300 nucleotides, and it is complex with the several proteins. So it's a protein plus RNA, hence it is known as the the small nuclear ribonuclear proteins, okay, or ribonucleoproteins. They're also known as the SNRPs. Okay. So they have got 3 roles in splicing. First is to recognize the 5' splice site, the branch site, and the 3' site. Okay. And इसी बीच में they need to bring those sites together so that they can catalyze, that is by cleavage and by joining the reactions. And to perform this function, they need to have an RNA RNA interactions, that is between your pre-mRNA and the RNA of the spliceosome, and of course the RNA protein जो आपका ribonucleoprotein है, and the protein protein interactions, so that they can carry out the catalytic or the cleavage and rejoining activity of the RNA, which is done by the protein part. Okay. Now, if you talk about protein cases, so there are different proteins involved in bringing the sites together. So if you remember in the previous figure, so we need to bring this side and this side together, right? So this bringing together is only possible once you have got different proteins, and the protein-protein interactions bring these two different sides together. Okay. So now slowly we'll see how exactly the spliceosome machinery is functioning. Now this is where you will see you have got the RNA RNA interactions. Okay. Thoda sa important hai, because you need to remember over here like which one is forming a base pair with the which spliceosome machinery. So already we mentioned that we have got U1, 2, 4, 5 and 6. Okay, 3 nahi hai. So just cancel 3 and rest all 5 are belonging to the spliceosome machinery. Now you can see that exon 1 can interact with U1 also and U6 also. Can you see? Okay, it is the same sequence of exon, but it can interact or it can form a complementary base pairing with both U1 and U6. Okay, exon 1 is with U1 and U6. Now coming to the next, U2. If it can just kicked out one base, it can interact with U2. 2 with U2. Then also, again, U6 and U2, these two spliceosome part can also form base pairing because both of them are having the RNA, correct? So they themselves can interact with each other. Then we have got the other proteins BBP also, which can bind to exon 2. ठीक है? Now BBP का काम क्या है? To take this A out so that the U2 can bind to the exon 2. Yes, guys, I hope this is clear. I mean, I'm sure you won't be able to remember this right now, but the concept is clear. Like who is interacting with what? Should I repeat? Shruti, is it clear?
It's clear, but I'll have to watch the whole thing to get the context completely.
Correct. See, that's what, because there is so much to remember. And I have given the warning that there are so many proteins and so many names. It's just the benefit is that the CSIR people or the exam people, they are not asking the history behind finding these proteins. ठीक है, नहीं तो बहुत ही life difficult हो जाती. So you just have to remember the function of the protein and not the nomenclature and like कहाँ से उसका the discovery हुआ and all. Okay, so I'll just repeat. I'll just summarize this slide. Okay, so now what you have understood is that in the spliceosome machinery, the spliceosome proteins. Okay, so there are 5 are there, and each of them are made up of RNA as well as proteins. Okay. Now this is important. Proteins are required for the enzymatic activity of the cleavage and rejoining of the exons. Okay, RNA is required so that it can base pair with the required exon. Okay, now exon 1 is there. Okay. Now you must be thinking, how come each exon 1 and exon 2, all these will be binding, right? So the thing is that in the genes, when let it be any gene, let it be take Sibin virus, T antigen you can take, or you can take insulin ka gene, whatever, this much stretch will be present so that the spliceosome can interact. Okay, of course they will have the sequence, but the evolution is in such a way that all these important genes will be carrying this stretch of the information so that the spliceosomes can bind to that and bring those exons together. Okay, so exon 1 will be interacting with your U2 and your U6. Now why U2 and U6 also will come to know once we start doing, U2, U1. Once we start doing like understanding how the spliceosome machinery takes place. Now exon 2 is with the U2. Now in order to facilitate this interactions, BBP is going to kick off this A, and then it will help in the complementary base pairing with U2. Among all the 5 SNPs protein, U2 and U6 can also base pair. ये कैसे? Just because of the similarity in the base pairing. ठीक है? Okay. Now with this understanding, let's proceed. So what is BBP? So BP is nothing but it is a branch point binding protein. So there are certain non-SNRNPs which are involved in splicing. For example, U2AF. AF is for your auxiliary factor, यानी कि वो help करता है आपको. So U2 अभी हमने देखा, it binds to your exon 2. So this U2 is having your subsidiary proteins are there. Okay, U2AF. So it has got 65 unit and 35 unit. Okay, so these 65 and 35 unit will help in recognition of the sequences which is present in the mRNA. Okay, for example, U2AF, it recognizes your polypyrimidine PY tract, that is 3' splicide. सो अभी आपको पता है 3' स्प्लाइस साइट कहाँ पे है, करेक्ट? अगर ये हमारा mRNA है, so we have got over here the 5' splice site. Here A is your branch point and 3' splice site is present over here. So to this region, your U2AF will come and bind. Okay. Now, in the initial step of the splicing reaction, it helps on the protein, that is your branch point binding protein BBP, bind to the branch side. So branch side में आपका आ जाएगा BBP. Okay, and as I said before, BBP क्या करता है, वो थोड़ा सा A को खींच लेता है so that U2 can come and bind over here. Okay, now BBP is then displaced by the U2 SNRP. So जैसे ही उसने A को ऊपर kicked off किया, U2 will come and bind over there. Other proteins which are involved in splicing reactions include RNA annealing factors, which help load the SNRMPs of the mRNA and the other dead box helicase proteins. And all this dead box helicase proteins, they have got the APPase activity, because as we know, when the RNA RNA interaction is happening, so वहां पे भी आपका interactions बन रहा है. So later I need to break this interaction so that the RNA जो splice हुआ है उसको हम separate कर सकें. So for that we need dead box helicase proteins. Okay. Now all these are required so that the splice mRNA from the spliceosomes can be removed, and later the spliceosome machinery जो है वो disassemble हो सके, यानी कि वो normally free हो सके and back to the cytoplasm. Okay. Now let's see the steps now. So this is really very important. मैंने और किसी में इतना questions नहीं देखा है जितना the questions they asked from the spliceosomes, because there are different pathways, different ways by which the RNA splicing happens. There are total 3 are there. Okay. So out of which spliceosome machinery is the most important one. So please do remember and try to understand everything. Okay. So first we have got the mRNA is there. Okay. So this is known as pre-mRNA. Okay, now as I mentioned, U2AF will go and bind to the 3' splice side, correct? And BBP, that is your branch point binding protein, will come over here जहाँ पे आपका A है. U1 will come to your 5' splice side. Okay, now this BBP will be displaced by U2. तो ये भी हमें पता है, जैसी A को उसने ऊपर की तरफ कर दिया, या यहाँ पे जो भी नीचे दिखा है, अगर U2 यहाँ बाइंड करता है तो A ऊपर की तरफ जाता है. Anyways, so it is going to interact with the U2. Clear? Now this is called, oh sorry, I need to mention this also first. So this complex form, that is the recognition of first spliceosome machinery, it is known as the early complex. So it is E. ठीक है. So E में कितने proteins आपके involve हुए? Your pre-mRNA, U1, BBP and U2AF6535. Okay. Now this is important guys. ठीक है कि E complex में आपका क्या क्या present है. Now A complex में क्या present है? Nothing. Just BBP replace करके हमारे पास U2 है. This is your A complex. Clear? Shall I proceed further?
Yes, ma'am.
Okay. Okay. Now I should mention here also that U2AF6535, 35 है जो आपका 3' splice side को bind करेगा, और जो 65 है, okay, this U2AF65 है जो आपका BBP को bind करेगा. ठीक है. This is also important because they do ask what is the arrangement of the U2AF. Okay. Don't worry, we'll summarize also. Now U2 आया, हमने वहां पे डिस्प्लेस किया, BBP को डिस्प्लेस करके हमने U2 डाल दिया. ओके. नाउ द बेस पेयरिंग बिटवीन द U2 एंड द ब्रांच साइड इज सच दैट the branch side A is extruded from the resulting stretch of double helical RNA as a single nucleotide bulge. तो जो bulge हमने देखा. Okay. So this is how so this A residue is then unpaired, and this is how now it is available for the 5' splice site. So अभी वो ready है हमारे पास. It is readily available for the attack now. Okay. Now the next step is the rearrangement of A complex. So U2 bind होना मतलब A complex. Now E हो गया, A हो गया. Now A से हम जाएंगे B complex में, ठीक है? So in this step, what happens is all the three splice subunits are there. Okay, they will come together. So if you have seen in the figure, so we have got 4, 5, and 6. Oh, sorry. 4, 5, and 6 splices are coming together. 1 ho gaya, 2 ho gaya. We don't have 3. So 4, 5, and 6 are coming together, displacing your U2A of 65 and 35. Okay. Okay. Now this is called those three snRNPs are known as tri-snRNP particle. Obviously, right? There are 3 particles are there. 5, 4, 5, 6 are there. Okay. Now 4 and 6, out of which 4 and 6 are held together by the complementary base pairing, and 5 is more loosely associated through protein-protein interactions. So, अगर आपसे ये पूछा जाए in this slide, what the takeaway message is that the interactions or the tri-snRNP particle is associated together on what basis? So you can say that U4 and U6 are together because of the complementary in their base pairing, and 5 is associated with 4 and 6 with the help of protein-protein interactions. Obviously, both of the interactions are weak क्योंकि इसको बाद में displace भी करना है, ठीक है? So what is the takeaway message over here? You have got the E complex, A complex, right? Now in the A complex, you have got the bulge A side readily available for the attack now. So for this, we need this tri-complex, okay? Tri-snRNP particle is required, which is made up of 4, 5, and 6. Clear? Now let's move ahead. Okay. Now can you see U1 and U2 tha? Okay. Slowly, what they have done, this 4, 5, 6 have taken the place of U1, and U1 is being displaced. Okay. So when this is formed, it is known as the B complex. So with the entry of the tri-snRNP, the A complex is converted to the B complex. And in the next, U1 leaves the complex, U6 takes its place. Okay. Now can you see how nicely the 5' branch site is coming near to the, 5' splice site is coming near to the A branch side. Correct? Can you see it over here? Slowly it is following those steps. Now this requires that base pairing between U1 snRNA and the pre-mRNA be broken, allowing the U6 RNA to anneal with the same region. Okay, so this will complete the assembly pathway. Clear? See, यहाँ पे क्या हुआ था? U1 is interacting with exon 1. So when this interaction is there, it means what? It is the RNA-RNA interactions of U1 and RNA. Correct? So U1 का हटा के now it is interacting with the U6 का RNA. Okay, clear? Should I proceed? Now, so you saw in the previous figure, just follow the figure, okay? Just follow the figure. So now we have displaced the U1. Now in this, what we have in the B complex, we have got 4, 5, 6, and U2 is there. Now the 4 has been replaced. Only 5, 6, and 2 is there, but it has done its work. क्या हुआ? पूरा का पूरा आपका lariat form हो गया. The 5' splice site is being broken over there and bound to the branch site. So one phosphodiester bond was broken and another was formed over here. Okay. Here now, to do this thing, your U2 and U6 were forming the complementary base pairing. And this part where U2, U6 are formed together, it is known as your C complex. Okay. Is it clear? Then next step. So यहाँ पे हमारे पास क्या है? The free 5' site is there. The free OH is over here, right? Over here. This will attack over here. What will happen then? Normally, so this gets released and I get all my exons together. Clear? Are you able to follow, Tanvi, Shruti?
Yes, ma'am. Yeah.
Okay, cool then. The thing is, you just need to remember the procedure. Okay, अगर आपने ऐसे भी याद किया कि let's like summarize. I'll tell you how, then you can remember. Okay, so this is, I don't think the figure is clear, but it is the same figure which I have taken in the bigger images and all. So just understand that there is a RNA. Okay, so let us just summarize what we have just discussed now. Okay, so first you have got the RNA, and you can see that to the 5' splice side, your U1 is coming. Okay, to your branch side, BBP is coming. This is your branch point binding protein. And then to the 3', it is your U2AF65, the larger subunit of the protein. And then you have got the 35. Okay. Now, next. So we know that this branch point binding protein is ensuring that this A gets protruded out so that it becomes available for the attack. Okay. And once this is being done, this is replaced by your U2. So U2 will come and bind over there. Next is that the entire tri-SNRNP particle is coming. That is made up of your U4, U5 and U6. Okay, they all coming together and replacing your U2AF65 pura hata denge. Now, slowly, with the rearrangement of the complex, U6 is going to replace your U1. कैसे करेगा? The complementary base pairing between the U1 and the pre-mRNA is being displaced by the U6 and the pre-mRNA. ठीक है? Now, U1 is gone. Slowly, what they're doing is now the U4, which is having the catalytic activity, is breaking a cut over here. Okay, making a cut over here. Then U4 is displaced. U6 is coming. This U6 is now making a base pairing with the U2 and making sure that the bond has been formed over here with the branch point. Okay. Later, the 3'OH is free over here at the first exon. This will have the nucleophilic attack on the phosphate which is present over here. So at the end, what I'm getting is exons are getting attached and the intron lariat, okay, the word used is lariat, is finally removed. So this is how your spliceosomes are functioning. Okay, clear? Thoda time lagega, but yeah, the more you see the figure and read about it, it will be clear to you guys. Okay, yeah. So 1 second. So I just wanted to add that the attack of 5' over here, the 3'OH attacking over here, which I was talking about, it's done by the U5. Okay, so that is done by U5. Just remember that. Okay, so there is this video I have got. I got it from this URL. Okay, so beautifully they have shown how this spliceosome machinery works. So let's watch this video. So you'll get an idea, like they have got the description also, like how exactly the spliceosome is functioning, as well as the animation. So nicely they have explained the spliceosome machinery for the RNA splicing.
As DNA is
the sound is audible. I mean, it's audible to you guys, the video sound?
Yes.
transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay. Clear? So now I hope you got the idea कि कैसे cell के अंदर after the transcription is happening, how the RNA splicing is being done. Okay, so basically in all the eukaryotes, mammals, splices of machinery is used. There are a few examples where I'll talk about the two different types of intron splicing. That is totally different. Okay, so is it clear? Do you want to watch the video again? Clear, guys? Okay, so there's a small portion left which I'll be taking tomorrow itself. Now the question what I want to ask is like, where does this RNA splicing is happening? Yes? कहाँ पे हो रहा है? Any idea? Is it in the nucleoplasm or it is in the cytoplasm? The splicing is happening inside the nucleus or outside the nucleus? क्या लगता है? Any guesses?
I'm inside.
Yes, it is correct. It is inside the nucleus. Because in the cytoplasm, only your matured RNA will be transferred. Okay, so there is another, what to say, export import mechanism by which the matured mRNAs are taken outside in the cytoplasm. Okay, so but unfortunately it is part of cell biology in which I will be taking. So that's why I used to say mole bio, cell bio. At the end only you will be able to imagine completely कि कैसे होता है. ठीक है? I mean, if I include over here, the story will go somewhere else. That's why I'm just keeping as part of cell bio over here, because in the cell bio, we are going to study how the proteins, चाहे वो mitochondrial protein हो या nuclear protein हो. So we know that the protein translation happens in the cytoplasm, but the nucleus is also full of proteins. DNA polymerase, RNA polymerase क्या है? ये सारे proteins ही तो हैं, enzymes ही तो हैं, right? So इसका मतलब cytoplasm से इनको nucleoplasm में लाना है. और कुछ ऐसे हैं जहां पे आपको न्यूक्लियस से साइटोप्लाज्म में ले जाना है. फॉर एग्जांपल दिस योर मैच्योर्ड mRNA दैट यू हैव टू टेक इट फ्रॉम न्यूक्लियस टू द आउटसाइड द साइटोप्लाज्म. तो ये पूरा जो एक्सपोर्ट इंपोर्ट का जो मैकेनिज्म है, इट इज क्वाइट ट्रिकी. And again, you need to remember again a lot of proteins. So just not including over here. So let it be the part of cell bio only. Okay. So I hope this is clear to you guys, like how the splices of machinery is happening. It's a really very clear and beautiful video. Okay. So I'll be stopping here now.